Good morning, children. Welcome to class 11th biology once again. Children, in a previous videos, we were discussing about chapter number 8, cell the unit of life, and we have discussed about cell membrane. Okay, now today in this video, we will cover cell wall, endoplasmic reticulum, Golgi apparatus and lysosomes. Okay, these are the endoplasmic reticulum, Golgi apparatus, lysosomes. These are membrane bound cell organelles. Okay, but start with cell wall first. Cell wall, you know very well, that is the outer covering, that covers the plasma membrane of fungi, plants, bacteria. Okay, so we can say that cell wall is non-living rigid structure that form an outer covering for the plasma membrane of fungi and plants. So basically the first structure that are found in the cell is cell wall, and it is consist in fungi by chitin. Okay, chitin is the polysaccharide that constitute the cell wall of fungi. Okay, and in plants the cell wall is made up of cellulose. Okay, children, if we're talking about the bacteria, the bacterial cell wall is made up of peptidoglycan. Okay, so basically the main function of the cell wall is give shape to the cell and protects the cell from mechanical damage and infection. Okay, it also help in cell to cell interaction and provides various barriers to undesirable macromolecule. Those macromolecule which are not suitable for the cell, so the cell wall that protect the cell from the undesirable macromolecule. Okay, clear about this? Cell wall, that is outer covering. In fungi it is made up of chitin, in plants it is made up of cellulose, in bacteria it is made up of peptidoglycan. Functions of cell wall, that is it gives the shape to the cell, it protects the cell from mechanical damage and infection, it also help in cell to cell interaction and provide various barriers to undesirable macromolecule. Okay, clear about this, children? So algae also have cell wall, and that is made up of cellulose, galactans and mannans. Okay, while the plant cell wall is made up of cellulose, hemicellulose, pectins and proteins, and the cell wall of young plant, that is primary wall. Okay, the cell wall of primary young plant, that is primary wall, and it is capable of growth. Okay, it is capable of growth which gradually decreases when the cell become mature. Okay, the primary wall help in growth, but it is decreased when cell grow, and after that the secondary wall is formed on the inner side. Okay, towards membrane. Clear, children, about this? Cell wall, basically primary cell wall form first time and it help in growth, but gradually it is decreased and the secondary wall is formed inside the cell, and this secondary wall play important role in protection, damage and give the shape to the cell. Okay. Now children, a layer that is middle lamella, that is mainly of calcium pactate, which holds the different neighboring cell together. Okay, the neighboring cell that are held by the middle lamella. So the cell wall and middle lamella may be traversed by plasmodesmata. Okay, the role of plasmodesmata, that is, it connects the cytoplasm of neighboring cell. Okay, so this is about cell wall that we have discussed. The main points we recall once again. The cell wall, that is outer covering, it give the shape to the cell, provide mechanical support, protect from mechanical damage, provide various barriers. Okay, and in different organism, the different polysaccharide are constitute the cell wall. Okay, in animals cell wall is absent. Okay children, now the endomembrane system. Okay, endomembrane system, that is consist of endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. Okay, so in this video children will cover endoplasmic reticulum, Golgi apparatus and lysosomes also. Okay, so start with endoplasmic reticulum. Endoplasmic reticulum are discovered by K.R. Porter. Okay, with the help of electron microscope, the K.R. Porter discovered the endoplasmic reticulum. Okay, so basically in endoplasmic reticulum, the network of tiny tubular structure that is scattered in cytoplasm. Okay, I'm showing a picture in front of you. This network of tiny tubular structure, this that is scattered in the cytoplasm. Okay, and these tiny tubular structure is called endoplasmic reticulum. Okay, clear about this children? So endoplasmic reticulum divides the intracellular space, okay, the structure inside, okay, the cell are divided by the ER, that is endoplasmic reticulum, into two distinct compartments. Okay, that is luminal compartments and extra luminal compartments. Okay, luminal, that is inside the endoplasmic reticulum, and extraluminal, that is outside the endoplasmic reticulum. Okay, so the space that is divided by the ER into the luminal, that is inside the endoplasmic reticulum, and extraluminal, that is outside the endoplasmic reticulum. Okay, so children, endoplasmic reticulum often show the presence of ribosome on their outer surface, and these ribosomes are play a very important role in protein synthesis. Okay, ribosomes that are found on the surface of endoplasmic reticulum, and they play important role in protein synthesis. Okay, so the endoplasmic reticulum bearing ribosomes on their surface is called RER, that is rough endoplasmic reticulum, and those endoplasmic reticulum do not bear the ribosome on their surface are called smooth endoplasmic reticulum. Okay, rough endoplasmic reticulum, they have the ribosomes, but the smooth endoplasmic reticulum, they do not have the ribosomes, that's why they appear smooth. Okay, so clear? Now, children, uh, due to the presence of ribosome, the rough endoplasmic reticulum RER frequently observed in cell actively involved in protein synthesis and secretion. Okay, so because ribosomes are there, so they help in protein synthesis. Okay, because the ribosomes function is protein synthesis. So if they found on the surface of RER, then definitely RER help in protein synthesis and their secretion. Okay, clear about this, children? While the smooth endoplasmic reticulum, they do not have the ribosome, so they will not play role in protein synthesis. Instead of protein synthesis, they are the major site for synthesis of lipid. Okay, in animals, cell lipid like steroidal hormones are synthesized in SER, that is smooth endoplasmic reticulum. Okay, clear about RER and SER? RER, that is rough endoplasmic reticulum, they help in protein synthesis, and SER, they help in synthesis of lipid. And also in animals, they synthesized steroidal hormones. Okay, this is about endoplasmic reticulum. These are tiny tubular structure that are scattered in the cytoplasm. Okay, next, that is Golgi apparatus or Golgi body. Golgi apparatus are discovered by Camillo Golgi in 1898. Okay, the scientist, that is Camillo Golgi, discovered the Golgi apparatus in 1898. That's why they have given the name Golgi apparatus or Golgi bodies. Okay, so the Camillo Golgi observed that some stain reticular structure near the nucleus. Okay, and later they have given the name Golgi bodies after discovery. The Golgi bodies basically they have flat disc shape cisternae. The size of the cisternae, that is 0.5 micrometer to 1.0 micrometer, and these are parallel to each other. Okay, I'm showing the picture of the cisternae. Cisternae, uh, these cisternae that are found near the nucleus, that is observed by the Camillo Golgi in 1898. Okay, so size, that is 0.5 to 1 micrometer in diameter. Okay, now children, the varied number of cisternae are present in Golgi complex. Okay, and the Golgi cisternae are concentrically arranged near the nucleus. If the cisternae that are arranged near the nucleus with convex cis phase, okay, so if we see the phase near the nucleus, the phase that is convex cis phase, and in the opposite side of nucleus, the concave trans phase or maturing phase, we can say that. Okay, by the use of diagram you see that this is near the nucleus, so that's why we can say that the cis phase. Okay, and if they are found opposite side of the nucleus, that is trans or maturing phase. Okay, convex cis near the nucleus and concave trans is opposite to the nucleus. Okay, and concave trans is also called maturing phase. Okay, this is structure of the Golgi apparatus. Now the functions of Golgi apparatus, children. The Golgi apparatus have important function in the packaging of the proteins. Okay, so basically proteins are packaged by the Golgi apparatus and they modify also. So modification and packaging of proteins takes place in Golgi apparatus. Okay, and Golgi apparatus is also important site for the formation of glycoproteins and glycolipids. Okay, two main function I told you. First one, that is packaging of proteins and their modification. Second function, that is, children, it is the important site. It is the important site for the formation of glycoproteins and glycolipids. Okay, so this is about Golgi apparatus that we discussed. Now last topic, that is lysosome. Lysosome is known as the suicide bag. Okay, lysosomes is called suicide bag. Okay, and they help in digesting. Okay, see about lysosome, children. Suicide bag, and these are membrane bound vesicular structure formed by the process of packaging in Golgi apparatus. In Golgi apparatus, when they package the protein molecule, they modify the protein molecule, their lysosomes are formed. Okay, and the lysosomes that are rich in almost all type of hydrolytic enzyme. Okay, so we can say that the various enzyme, that is lipases, proteases, carbohydrases, they are found in lysosome. Okay, and these enzyme, they active at acidic pH. Okay, and help in digestion. Okay, so basically the carbohydrates digest the carbohydrate, proteases digest the proteins, lipases digest the lipids. Okay, so this is about lysosome. Lysosome, I repeat once again, that is suicide bag. These are formed during packaging of protein molecule in Golgi apparatus, and lysosomes have rich in almost all type of hydrolytic enzymes, lipases, proteases, carbohydrates, and these enzymes activate at acidic pH. Okay, the question may be asked, at which pH the hydrolysis enzyme will activate? So on acidic pH these enzyme will active and they are capable of digesting carbohydrates, proteins, lipids and nucleic acid. Okay, clear? So children, that's all for today. I hope you have learnt what we have discussed. If any problem, we'll discuss in live classes. Thank you.